sorry you guys, as would be typical for my generation, I got a phone call and it cut off my video and I didn't know what to do. So what I was saying at the very tail end was, look at how small these things were, okay? And the comparison was that number 10 right here is a single molecule of hemoglobin, which of course is the oxygen carrying molecule in all of your red blood cells. So, yep, you're dealing with organisms that are incredibly small. So here's some examples. The other thing I wanted to show you is that even when these were seen under the electron microscope, they still had a really unique appearance. So when you look at these, they're very crystalline in structure, and that's because of the capsule mirrors that are forming the capsid. You know, they all fit together like little puzzle pieces. You can see it a little better over here. So scientists were like, wait a minute, these aren't cells. They don't look like cells. So it took them a while to figure out the mechanism of, you know, are these infectious and how are they doing it? Excuse me. As an example though, this is what Ebola looks like. So Ebola is a long cylindrical virus, but then it kind of curls itself into, it kind of looks like a candy cane structure to me, but that's what Ebola looks like. Okay, so we've talked about this a little bit, but so these regular repeating subunits that make up the capsid are called capsule mirrors, and that's what gives them their very crystalline appearance. So remember, if you just have a capsid but you don't have an envelope, you're referred to as a naked virus, whereas viruses that have envelopes are called enveloped viruses. And the core of the virus, its genome, is either DNA or RNA. So here we've seen this figure before. So the virus on the left would be a naked virus because it doesn't have an envelope. The virus on the right would be an enveloped virus. Now, naked viruses can also have spikes. So you can be, naked just means not having an envelope. It doesn't refer to the spikes. So the capsid is made up of protein, and it is constructed from these identical subunits that are called capsule mirrors, and that's what gives it its very crystalline type of appearance. Let's see, once again, this is showing a cylindrical virus. So here you have the nucleic acid, and then you have the capsid on the outside. And I believe this is HIV. So HIV is roughly spherical, but you have the envelope on the outside, and then you have spikes, which aid in attachment. Okay, this is our little buddy adenovirus. So adenovirus causes upper respiratory infections, and a lot of you, I'm sure you've experienced this. So just want to show you have a little moral support over there. Hey Gert, how's it going? So if you've ever woken up in the morning, and by the way, those are allergy coughs. Those are not anything related to COVID-19, just so you know. If you've ever woken up and you have that little tickle in your throat, like, oh crap, I'm coming down with something, and then about 8 hours later, it feels like you're gargling shards of glass, you can usually thank this guy, okay, adenovirus, because it's causing upper respiratory infections. So this is an example of a virus that's naked, but it does have spikes on it. Okay, here's some electron micrograph photos. So these are enveloped viruses inside a cell. So you have the capsid shown in red, the DNA cores in green, and then the envelope, which is pretty thick, is shown on the outside in blue. And this is what happens on the right when a virus goes through its replication cycle. So it hijacks all of the cellular machinery, meaning all of the nucleotides, the polymerases, the ribosomes, the transfer RNAs, your amino acids, to make more copies of itself. And then eventually the cell just kind of blows up and it releases all of those viruses where they can then float over and infect adjacent cells. So that's how it goes through its cycle. Okay, this is an example of what we call a bacteriophage, and I'm glad it only infects bacteria because it creeps me out just looking at it. It kind of has a spider appearance. It reminds me of daddy longleg spiders, but these are its legs, and they are what will allow it to attach to the bacterial cell wall, and then the virus literally sits down, and it has what are called tail pins, and there are some enzymes in the tail pins that allow it to kind of just eat through the bacterial cell wall. And then, for lack of a more professional term, it basically poops its nucleic acid into the bacteria, and then that kind of starts the whole process. So these can be useful, however, in delivering medications. They've been used in a lot of viral research. We can work with them in the lab because you don't need a sterile cell culture facility to be able to do this. You just need bacteria. Okay, so once again, these are examples of enveloped viruses. These are examples of naked viruses. Naked viruses can still have spikes. Okay, the envelope. So a lot of viruses, and really this is super smart on their part. So when the cell is full of viruses and it's ready to explode, a lot of viruses that are enveloped will actually just steal part of the cell's membrane and then use it as their envelope. Now, the reason that is so smart is because it's a perfect disguise, right? Your immune system is going to look at that and say, well, that's me, right? It's showing some of the normal, proteins that would belong to one of my cells. So I don't have to worry about that. That's supposed to be here. So it's a great disguise. Now, eventually, what gives viruses away is that the proteins that are in that envelope are going to be replaced with viral proteins, okay, like those spikes that allow an attachment. So it's a great short-term disguise. It's not going to last forever, but hey, if the virus can buy some time that way, that's a really smart mechanism to use. So when we talk about functions of the capsid and the envelope, so one of the things that the capsid does that benefits the virus is it's a nice hard outer coat that protects the nucleic acid core in the center, and that's where the viral genes are. So obviously really important because those genes are going to direct the making and synthesizing of new viruses. It also, in terms of the envelope and spikes on the capsid, that's going to help bind to a host cell and get that virus into the cell in the first place. Okay, so those are two things that, they're helping out the virus. What helps us, if we're talking about us as a host, those viral capsid proteins can be used to produce vaccinations that can then immunize us and get us to stimulate and produce antibodies that will protect us when that virus is introduced into us in the first place. So the best thing you can do to protect yourself against really deadly viruses is to get vaccinated, okay? Anti-vaxxers, oh my God, don't even get me started. Not gonna go down that road quite yet. But when viruses invade you, if you can have antibodies in your system that suck up that virus and basically neutralize it before it ever gets to your cells, that's your best way to protect. Because once the virus is inside your cell, there are medications that can help slow it down, but you would have to destroy your own cells to get to the virus. And you don't want to take medications that are going to blow up your own cells, right? So a lot of antivirals, like, say for influenza, like Tamiflu, I've never taken them, but I've talked to friends and coworkers that have, and what they tell me is it makes the flu suck less. So, instead of having a flu that knocks you on your butt for 10 days, it may knock you on your butt for maybe 5 to 7 days. So it's not like an antibiotic that's going to actually destroy, hopefully that organism. It doesn't happen with viruses because they're hiding and doing what they have to do inside our cells the majority of the time. So yes, vaccination is your best bet against protecting yourself against a lot of these deadly viruses. So the genome, remember the definition of the genome is the sum total of the genetic information carried by an organism. And viral genes are really, really small. And I'll give you some numbers in our next lecture. I just need to look up those numbers. But it's really compact. They carry very, very few genes, only the ones necessary to get the virus inside your cells and then to hijack your cell. That's it. Okay, so they're not going to carry with them, genes to, make their own polymerases. Why do that when they can use yours? Why carry genes to make amino acids? They don't have to. You have all the amino acids they need. So viral genomes are very, very small, and that's why viruses can be small. And of course, remember this, viruses either have DNA or RNA genomes, but never both. Okay, so let me stop there. We'll pick up here in our next lecture. All right, thanks.